My presentation is going to be on development of a new liposomal irinotecan formulation and its immune activation potentials. So irinotecan or CPT11 is currently being used in first and second line colorectal cancer treatment. Its use is often associated with dose-related GI toxicities, and the drug undergoes the interconversion process at pH 7 in the blood compartment, which inactivates the drug into its carboxylate form. So these features of the drug has led to the idea of putting it into some kind of drug delivery systems, such as liposomes. So our lab had previously developed a liposomal CPT11 formulation called Irinotecan, which relies on the entrapped copper inside the liposome to actually retain the CPT11 with the liposome and also to stabilize the drug in its active lactone form through this interaction here. However, the preparation method used in Irinotecan was not optimal because majority of the copper was lost during the loading process. So my idea was to come up with a better method to prepare this formulation, which retains the copper during the loading process, and we hope to achieve some sort of improvement. So as you can see, we are calling this new formulation high-C formulation. So with the copper retained with the liposome during the loading process, we actually be able to achieve a higher CPT11 loading capacity, where we can achieve a maximum drug-to-lipid ratio of 0.6, improved from 0.4 in the Irinotecan formulation. And also, when we analyze the formulation by cryo-EM analysis, we actually see the drug form a gel-like precipitations inside the liposome, which is different compared to Onivate, which is an alternative liposomal CPT11 preparation. And also, when we store the formulation at 4 degrees Celsius, the high-C formulation is actually more stable than the Irinotecan, which had precipitation issues and the copper leakage. So next, we went on testing whether our formulations is going to be more effective in vitro in the cytotoxicity assays against the mouse colorectal cancer cell lines. So we seed CD26 and MC38 cells in 384 well plates and treat with either our formulations or free drugs and expose the cells to the drug at different concentrations at 4, 24, and 72 hours. And as you can see here, longer exposure of the cells to the drug is actually more beneficial, but more importantly, our formulation is more effective at killing those cancer cells. The IC50 value is around 4 times lower for our formulation. And then we want to see whether this in vitro efficacy improvement is going to be translate something in vivo. So we did this efficacy study where we inoculate the cells subcutaneously and give the dose at 40 milligram per kg every other days for 4 injections in total. So 1 week after the final injection, we actually observe a significantly delayed tumor growth in the high-C treated group compared to all the other groups. Of course, this delayed tumor growth has led to improved survival times. So it is shown in literature and also in our lab that the efficacy improvement associated with liposomal irinotecan formulations primarily has to do with the improved exposure of the drugs to the tumor, and the encapsulation of the CPT11 into liposomes did exactly that and stabilized the drug after injection. So we start to wonder whether the activity of our high-C formulation is governed or at least related to the immune systems. And we propose 2 different mechanisms. One is that induction of tumor vascular normalization, and another is the induction of immunogenic cell death, or ICD, which I'm going to be focusing on today. So what is ICD? It is a specific type of cell death, and it triggers the immune response. So cells undergoing the ICD process will release damage-associated molecular patterns into the surroundings, such as increased ATP secretion or HMGB1 secretion from the nucleus, or the cells will actually expose keraticulin on their surface undergoing ICD. And then those dying cells will then attract antigen-presenting cells, or APCs, in the tumor microenvironment, which subsequently take in those dying cells and then presents the antigens on their surface, and then activates T cells, which subsequently kill off the tumor. So as I mentioned, there are several markers that you can use to assess the level of ICDs in response to your treatment. So the one we chose here is the HMGB1, where we seed the cancer cells in 24-well plates and treat it with either the free drug or formulations for 24 hours, and then harvest the culture media to analyze the soluble form of HMGB1 present in the media. So as you can see here, the CD26 and MC38 cells both respond really nicely to our high-C formulations. So compared to the free drugs, they have increased level of HMGB1 secreted into the surroundings at 2 different concentrations. And also to validate that this ICD-inducing effect is not specifically towards only mouse CRC cell lines, we tested in 41, which is a mouse breast cancer cell line, and also we use HCT116, which is a human colorectal cancer cell lines. And then again, we observed something similar, that the high-C formulation is better at inducing HMGB1 secretion from the cells compared to the free drug. And next, we want to test whether this ICD-inducing effect is going to be translated to something in vivo. So we designed this vaccination study where we treat the cells ex vivo with our liposomal formulations, leave for 24 hours, and then inoculate those dying cells into the left flank of the syngeneic mice, and then re-challenge them after 7 days with healthy tumor cells and monitor for the tumor growth on their right flank. So, and ideally, if you have the cells undergoing ICD recognized by the antigen-presenting cells, they will then have a slowed tumor growth compared to untreated groups. So, of course, there's no obvious tumor growth on the left flank of the animals in the treated groups. And when we take a look at the treated groups and compare it with the untreated groups, we actually see that the mice inoculated or vaccinated with our high-C treated tumors, they actually have significantly delayed tumor growth in the high-C treated groups compared to the untreated ones. And in the CD26 models, we have 5 out of 8 animals stay tumor-free long-term after 3 weeks after the last animal in the untreated group went down. And we also see a slight improvement in the MC38 models, but not as significant as what was in the CD26 models. Some of the future work directions, we want to demonstrate that the cells treated with the high-C actually can activate the APCs, and also investigate whether our formulation is going to be synergistic when combined with some sort of immunotherapies. So with that, I would like to thank my supervisory committees and all the lab members who helped on this project, IDP and PharmaCore of MMIN. Special thanks to Nancy and Nico here, you're the best, and all the funding agencies. Since I still have the mic, I will take the final 20 seconds to say we have 2 really brilliant young trainees, Kat and Janelle, who I have worked with, and they are in their final undergraduate years looking for opportunities to get enter into the industry or grad school. So if anyone wants to engage, I'm happy to do the introduction. So thanks.
Thanks, Kevin. I can turn that off. Even with the extra time, you still had a whole minute. Way to go. We have plenty of time for questions. Yep.
We all know that Inducer is a prodrug, which is not the actual drug. Did you include in your studies any experiments with the actual drug, which is N-38?
Yeah. We did not try to encapsulate SN38 in this formulation. However, we did pharmacokinetic studies to analyze the SN38 levels in the blood following the injection of our formulations. It's actually showing an improvement over the free drug as well.
Other questions, Urs?
So how could you do this immunization approach in patients?
This immunization or the vaccination study we designed here is to actually validate that the cells that's treated with our formulations actually undergoing some sort of cell death that will trigger the immune response. We are not particularly looking into, developing a cancer vaccine with this formulation here. However, I do know that there are other studies that actively investigating using the dendritic cells here that's recognizing the dead cells to develop into a cancer vaccine project.
Cool, thanks. We got a question in the back.
So if I understand correctly, do I think that the immunization effect I see can also be done with Irinotecan?
No, can it be done? Is it just due to the drug?
It's just due to the.
It has nothing to do with the immune system. The effect you're seeing is due to the drug.
It's a really good question. Here I'm gonna say that the answer is pending because there are actually other factors that come into play when you are doing those kind of vaccination studies. For example, how the tumor are actually forming, how the cells actually infiltrate into the tumors. The list sort of goes on. But in my opinion here is we actually designed the study such that the majority of the cells are actually dying when we inoculate it. So the viability is around 50%, 30 to 50% when we inoculate it into the mice. And then during the process, we actually washed out the drug. So it's only the cells that's being inoculated into it. So in my opinion, I think that the dying cells is the only kind of test articles in that scenario.
Good answer. Star, last question.
How does your liposomal drug kill cells
In vitro.
In vitro, yeah. So I think I'm taking a guess here. So I think one of the reasons that the liposomal formulation actually stabilize more drug or more encapsulated CPT11 in its active form and slowly release into the surroundings. So over a long period of time, you will actually have a higher exposure of the cells to the active lactone form of CPT11. So that will actually improve the in vitro cytotoxicity of the liposomal formulation compared to the free drug, where you gave it and then majority of the drug will be inactivated into its carboxylate form in 30 minutes, 1 hour. So.
Good answer. That was what I was going to say. Perfect. Thanks very much.